Welcome back to our session dealing with the genetic code, translation and protein biosynthesis. After we have learned a bit about general properties of the genetic code, let us try to use the genetic code table to find out what combinations of bases actually produce certain amino acids. So what you can see here is the table. In order to use it properly, you need to remember that each triplet in the genetic code is read from the 5' to 3' direction. So the first base here is the 5' base, followed by the middle base, and then the last base is the 3' base. So let us have a look at an example. In order to identify the codes coding for arginine, we need to start at the first letter. So first letter here is a C, right? You need to go to the left and then you will find that the first letter is a C. The second letter here, you need to go up, is a G, right? And last but not least, if you look at the 3' third basis, then you will find that U is one possible third base producing arginine. So the first code we have identified here is CGU, with C being the first base, G being the second base, and U the third one. As I explained to you earlier, the genetic code is degenerated. That means that there are different possible combinations producing the very same amino acid. So in case in arginine, there are at least 3 more possible combinations. However, the first letter and second letter are gonna be the same, right? So if we go to the left again, then we will see that C is still in there. The second letter, G, remains unchanged. However, the third base is gonna be a C, right? This is the second possible combination. If we follow this exercise further, then we will end up with CGA, and the final combination is CGG. Those are the codes for arginine. What I would like you to do now is to use this genetic code table to tell us what the appropriate codon for leucine is. And in order to do so, I would like to ask you to submit the form which will pop up in the video shortly. Right, so I hope that all of you came up with the following combinations. So what you need to do here is to identify the abbreviation for leucine. This is obviously here. Then if you go to the left to identify the first letter, you will find that this is actually a C. So the first code is C. Second letter, if you go to the up, is U. And the third letter is gonna be U, okay? So the first possible combination is CUU. If you follow the exercise similar to what we have done for arginine, then you will come up with the following codes: CUU, CUC, CUA, and CUG. In addition to the code table, there is actually an easier and more modern way to identify the codes for certain amino acids, and this is the codon wheel. And from my point of view, the codon wheel is even easier to use, and it's more useful, at least for me. So what we are going to be doing now is to do the very same exercise we have done with the genetic table with the codon wheel. So we would like to find out the combination for arginine. So what we have to do now is basically to start at the 5' end. We need to find the arginine. Arginine is obviously here. And again, we need to start at the 5' end. And the first letter is C. The second letter is gonna be a G, followed by 4 different possible combinations. So the first possible codon is going to be CGG. Second one is going to be CGA. Third one, CGC. And the fourth one is going to be CGU. So similar to the exercise before, I would ask you to use the codon wheel and come up with the possible codons for proline. I hope that all of you have been able to come up with the following codes: CCG, CCA, CCC, and CCU. Now, please do the same exercise to find out the codons for valine. I hope that all of you have been able to successfully identify GUU, GUC, GUA, and GUG as possible codons coding for valine. The last exercise I would like you to do using the codon wheel is to identify the stop codons. So what you have to do now is to find the stop codons and then just start at the 5' and come up with the correct codes initiating stop of the translation. Obviously, there are 3 possible stop codons, and the stop codons here are UGA, UAG, and UAA, right? So if we look at this stop codon, we need to start with U. The second letter is gonna be A, and then A. Next one is UAG, and the last subcodon is UGA. Brilliant. So if we now look at the genetic code at the DNA, then we need to remember that mRNA molecule resulting from the genetic code on the DNA is actually gonna be antiparallel. Okay, so if the code on the DNA is TAC, then the RNA, complementary RNA, is gonna be AUG. If we now add tRNA, then the direction changes again. So we have UAC, which is quite similar to the code on the DNA. And those are basically the rules you need to keep in mind. The mRNA molecule is antiparallel compared to the DNA molecule, and in addition to this, tRNA codons are antiparallel to the message on the mRNA. And then we have the amino acids attached to this sequence. So in order to practice this a bit more, I suggest that you engage with an interactive online learning activity, which is provided by the University of Utah. So if you follow the link I have put in this slide and also on Blackboard, you will end up with a very, very interactive exercise, actually helping you to remember those base pairing rules, the polarity of nucleic acids involved in this process. And again, this is a very, very useful tool that is going to be good for you for the exam. With this, I would like to end up our session number 2, and I'll see you in session number 3 very shortly. Thank you very much for your attention.